Associate Professor at University of Arizona. Again, I already did some introductions, so we'll
Arizona State.
Arizona State.
You on that one. Very important to us.
So you're on.
All right, thanks. Great. I'm really honored and touched that Jose asked me to speak here today. I've known Jose for years through the community of evolution and cancer. I'm going to show you some of his legacy in that context. So the first bit is important and probably well known to everybody, which is that neoplasms evolve, cells within tumors evolve. This is the famous paper from that of Peter Knoll from 1976. And in fact, we talk about cancer as really an example of multi-level selection, a multi-level evolution. And part of my training as an evolutionary biologist, and evolutionary biologists love this stuff because you can get evolution happening at different levels. Then, of course, cancer kills people, kills organisms. Oh, hey, Doug. So it has a selective effect on the organism level, right? So there's selection and over evolutionary time to build up cancer defenses, cancer suppression mechanisms, which have allowed our ancestors to reproduce and benefit us in terms of all the cancer tumor suppressors we have and things like that. There's an interesting level at the level of epithelial units like colonic crypts that Jose has worked on. And those crypts, and this is a famous paper by John Cairns in 1975, showing that the tissue architecture of crypts actually constrains clonal expansion. So mutant stem cell might take over a crypt, but it may have a hard time getting out of that crypt, and so prevent a clonal expansion. And we don't actually know much about what causes a epithelial unit to divide or die. We don't really know much about the evolution at that level, but it's actually, some of our models show that's critical to the timing of parthenogenesis. So that's something I think we'll have to work on more in the future. Of course, what I just showed you is that evolution happens at the cell level, and in this case, selection at the cell level selects for cancer. So you have selection against cancer at the organism level, selection for cancer at the cell level. All the hallmarks of cancer are phenotypes that benefit the mutant cell that has that phenotype. So ignores growth suppression signals, generates its own growth signals, suppresses apoptosis, on and on. There's also interesting dynamics happening at the gene level. So you can get mobile genetic units, elements that can copy themselves within the genome without even the cell dividing, and so the number of copies of that gene will increase over time. There's natural selection at the gene level, and of course we know that these get demethylated in cancer, and we think that it's one of the contributions of genetic instability that drives a lot of the cell level evolution at cancer. So interesting interacting dynamics at these different levels, and in fact Jose has worked at every single level here. So I wanted to highlight this. It's really impressive. Of course, his clinical work is at the organismal level. He's got this JTV paper looking at the epithelial units that Larry was just mentioning, looking at actually cooperation between cells. A famous paper by Ricard and Jose, which is actually how I first came across Jose's work, looking at cell level selection in cancer and genetic heterogeneity, which I'm going to talk more about. And then even working at the mobile genetic element level and demethylation with Paul Lizardi. So that's sort of the amazing spectrum and breadth of Jose's work. And another thing that really comes out in a theme of his work that's highlighted by the symposium is the way that he's combined observation and models, a theory, to really make novel predictions and to understand those observations. And these are both models from those two papers that are intimately bound up with the advances that he's made. Okay, so back to the cell level evolution. And this is a sort of a pathology story that I want to tell that derives from this problem. So we know that tumors evolve, but if you have a particular sample of a tumor and you go to a pathologist in the room, if your oncologist colleague asks you, well, what kind of evolution is happening in this tumor, right? It's a hard question to answer. What do you mean? We don't have a language with which to make distinctions between types of evolution in a tumor. And Del Shibata was thinking about this and pointed out that the problem we have in describing the kinds of evolution of a tumor is similar to the problem that doctors had with mental illness in the 19th century. At that point, we didn't have ways of making distinctions, of classifying, of categorizing mental illness. It was just people were crazy, people were mad in different ways. And the big advance then was the DMS, where regardless of its various flaws, where we started having a way of systematizing mental illness, drawing distinctions, making categories. if you can't make those categories, you can't make much progress. If you can't distinguish depression from schizophrenia, how can you do a clinical trial to help those people, right? So if we're going to do this for cancer, we can draw a variety of distinctions. We can look at the morphology, we can look at the molecular biology, and Larry pointed out some of the subclassifications in breast cancer we can make based on the molecular biology. But what about subclassifications based on how they're evolving, the evolvability? So what are the relevant evolutionary distinctions? So we had a consensus workshop in 2016 where we brought together a lot of the people that work on the evolution and ecology of cancer to try to work on this. Can we develop a framework, a classification framework by which to describe differences in the evolution and ecology of tumors? So here's one that I think the evidence is most strong for, which is the tumor heterogeneity, or we also sometimes call this genetic diversity. This is a figure from that paper with Ricard and José, where they looked at colorectal cancer, and the color coding is for different mutant combinations of mutations across 3 different loci, and really brought out the fact that tumors are genetically heterogeneous. Right? And this is key for at least 2 reasons. One is that heterogeneity is the fuel for natural selection. If there's no heterogeneity within a population, you can't get natural selection. One type can't be better than another type if there's only one type, right? It's also, of course, then the fuel for therapeutic resistance, right? Because our big problem in oncology, of course, is we come in with a very strong selective pressure, we kill all the sensitive cells, and we select for resistant cells. So that heterogeneity is going to be key to how a tumor responds. And one of the insights that Ricard and Jose had from this was based on this model, which is how can you get such heterogeneity that's interleaved, right? It's not one big yellow region, one big red region in this figure, right? They're interleaved. How can you get that? And from a relatively simple model, what they found was that actually just from the very growth of the tumor with these different mutant clones, they naturally tend to interleave. You don't have to posit anything special to get this dynamic. I want to draw a line from that to recent work from Andrea Sotoriva and Trevor Graham on what's called the Big Bang model, again, started in colorectal cancer, where they would take individual crypts from opposite sides of a colorectal tumor and find the same clone on both sides, but interleaved with other clones. So again, these clones are not growing as coherent, separate patches, but are interleaved. And they made a simple assumption of actually neutral evolution, so no natural selection happening, just a sort of big bang from the initial transformed cell, the fully cancerous cell, growing outwards. And if there was some cell movement initially, then you get a mixing of clones, and as they expand, you can see the 2 different clones on 2 different sides, right, mixed together with other clones. So essentially the same thing that Jose and Ricard saw in their paper years before. Now, one interesting observation that Andrea and Trevor made was that in the adenomas, you don't tend to see this. You don't tend to see the same clone on both sides. You tend to see the clones on separate sides. And that leads to an interesting hypothesis, which is this mixing may be a signature of the cells moving around in the early stages. And perhaps that's actually a sign of risk, right? If cells can move around a lot, they seem to be generating carcinomas and not adenomas, and also possibly more metastatic. Now, another really nice thing that came out of this is just neutral evolution, where there's no natural selection, is actually very predictable. So if a population grows, then you look at the very first cell division. If you have a mutation in that first cell division, over time, that mutation is going to show up in approximately 50% of the entire tumor. It just gets amplified by tumor growth. If a mutation happens in the second cell division, it's going to show up in 25% of the entire tumor, roughly, give or take some noise. So if a tumor is growing neutrally, you expect to see this characteristic frequencies, and it's sort of, let's see. You should expect to see this slope here with a few mutations at sort of 50% of the population, and many mutations at smaller percents, because they're happening at these twigs, not in the trunks. Okay? So the actual slope of these subclonal mutations tells you something about the dynamics of the tumor. In fact, if it fits if you have this characteristic feature of neutral evolution, it should have a characteristic curve, and so you can actually take samples and fit them to the predictions of neutral evolution to say, well, these glioblastomas really don't seem to be evolving neutrally, but these lung adenocarcinomas or stomach adenocarcinomas, even colon cancers, a lot of them tend to be growing neutrally. In fact, about 1/3 of colorectal cancers seem to have this characteristic neutral evolution. That's neutral evolution after transformation. So there's natural selection all throughout carcinogenesis to build up those hallmarks of cancer. But once it becomes a full-blown cancer, in a third of these tumors, there's no further strong signature of one clone having an advantage over another of getting natural selection. I already mentioned this intriguing possibility that came out of this work that maybe mixing is a predictor. But this diversity, as I said, really gives what we call in evolution evolutionary resilience to the population. If you have a lot of genetic diversity in the population, it can handle any selective pressure you put on it because there's going to be some mutants in there that'll be adaptive under the new pressure. And unfortunately for us in cancer therapy, that new pressure is our therapy, and you're selecting for its resistance. So that leads to the prediction that the amount of genetic diversity should be a good predictor of how the tumor is going to behave over time. This is in the context of precancer in Barrett's esophagus. And what we showed is if you measured diversity at baseline in the Barrett's esophagus tumors, it predicts the patients that are more likely to go on to progress to adenocarcinoma of the esophagus. This is based on a measurement called divergence. So that's not how many different clones you have, but how different genetically are the clones that are there. How long has it been since they had a common ancestor? But there are other ways of measuring it. It doesn't matter whether you measure driver mutations like P53 or P16 versus passenger mutations that are evolutionarily neutral. The amount of diversity predicts progression to cancer. Oops. And these measures of diversity are also robust to the statistic you use for quantifying diversity. So I was mentioning divergence. You can also just count the number of distinct genetic clones, or use an ecological measure of species diversity, which accounts for not just number of clones, but their relative abundance. All of them are highly significant predictors of progression to cancer in Barrett's esophagus. Then a couple of years ago, we analyzed the TCGA data. So 1,100 tumors across all different kinds of cancers, used Noemi Andor's method called expands for deconvoluting the clones within one sample, so estimating how many clones are just in the biopsy that was sequenced. And if you do that, the tumors that have more clones those patients have a worse survival than patients with fewer clones in their tumor. So now we've switched from predicting progression of cancer to predicting survival once you have cancer. And the underlying theory, of course, is the same. The more diversity you have in the tumor, the more chances you have, in this case, for resistance to therapy, or in the other case, for the next hit that will facilitate progression. Okay, so if we're going to quantify, if we're going to try to classify the evolvability of a tumor, one thing we'd probably want to do is measure the degree of genetic diversity. But that's not the whole story, because evolution is characterized by more than just the diversity in the population. And here's a little illustration, a schematic. So this might be a cell lineage of a tumor. It's got the exact same branching pattern as this cell lineage. This is time on the x-axis here. But this one is slowly accumulating mutations. This one rapidly accumulated mutations just in the last period of time. So same genetic diversity in these two cases, but different dynamics. You can think of it just maybe in terms of mutation rate. In fact, how the tumor changes over time and quantifying that, it's not clear what the best way to do that is. Maybe how diverged are the tumors, as I mentioned earlier, or their mutation rate, maybe whether or not they have a mutator phenotype, whether or not you're seeing hopeful monsters show up, which is an old term from evolutionary biology, just rather than small accretions of mutations, big jumps like you might get in a tripolar mitoses. And it's also an open question whether we should be measuring the phenotypic diversity or the genotypic diversity. This is a figure from our work where we actually were able to measure mutation rates in vivo in Barrett's esophagus given 2 time points. So if you can study a tumor longitudinally and take samples, you can use methods from evolutionary biology to infer, to measure the actual mutation rates over time. But it's open. We don't know yet whether those mutation rates are important clinical predictors. So the end result of our discussions in this consensus conference is that the 2 major parameters we should be looking at are some kind of measure of diversity and some kind of measure of change over time, which we describe as delta or D. And if you just had maybe thresholds for quantifying low versus high, you could get 4 categories of the evolvability of tumors. We call that the EVO index. But the evolution is not the whole story, of course. That's a very cancer cell-specific view. The microenvironment has a big effect on what's going to happen to the tumor, right? And the way I think about ecology is the ecology is a way of describing what are the selective pressures on the current population. And those selective pressures are, of course, are going to determine what happens next to that population. So if you ask an ecologist to try to start classifying or quantifying the ecology of a species, they generally split the ecology into the hazards that are faced by the species and the resources that are available to the species. This is the initial division we proposed. And here's some data. There's some work that we did with Yin Yuan at the Institute of Cancer Research in London, where we use an ecological measurement of the degree of interaction between two species. So it's called the Morosita-Horn Index. And if two species are interacting, then they're interleaved a lot. That suggests that there's a real ecological relationship between them. So Yin Yuan is a digital pathologist or develops digital pathology algorithms to quantify the locations of cancer cells, immune cells, fibroblasts. And then you can apply the statistic to describe how interleaved are the cancer cells and then the immune cells. And of course, it's not clear whether immune cells in general are bad for the tumor or good for the tumor. Of course, you can have T cell responses, macrophage responses can be pro or anti-tumor. But we apply this to a large, the Metabrick database of breast cancers. And what you can see is that when there was an interleaving of the immune cells with the cancer cells, those patients had better survival than when the immune cells were separated from the cancer cells. And this is particularly strong in the HER2-positive cancers, which implies that in general, in this data set, the immune cells were having an anti-tumor effect. There's some kind of predation, essentially, relationship between the immune cells and the cancer cells. This is, of course, not a surprise to anybody in the audience that immune response is important. And this other data, and this is not our work, of course. With just a 2-gene immune signature, you can get strong clinical results predicting survival across a lot of different cancers. So quantifying the hazards by something like the immune signature, I think it's going to be an important way of describing the ecosystem. Of course, there are other hazards that are faced by cancer cells. There are various toxins in the system. We don't know much about the role of the microbiome in affecting the ecology of cancer cells. And of course, our therapeutic interventions form dramatic hazards in their environment. Resources is one that I think we poorly understand at the moment. What are the limiting resources for a tumor? And all of us know that you can get regions of hypoxia and necrosis, so clearly oxygen at some point in some cancers, in some regions of a tumor, it can be limiting. But in general, there are periods where it's actually more limited by glucose or phosphorus. We don't really know. This is other work by Yin Yuan. Again, you can classify she takes an H&E image, classifies the cells into different types, and starts quantifying. In this case, she's looking at fibroblasts. Fibroblasts is potentially a form of resource for the cancer cells, providing some kind of support signal, and then developing some classification system. In this case, in ovarian cancer, there's other work looking at lactate in the cancer and showing that you have clinical prediction. So very much open question about what should be measured, but we need to develop some kind of measures of the resources and what's limiting for the cancers. So again, if you were to describe a threshold for high versus low resources or high versus low hazards to the tumor, you could get these 4 categories, different kinds of ecology, so which we call the eco index. Of course, you may find that you need more than 2 categories. But if you did just use 2 categories for each of these measures, you get 16 classes of cancer, which quantifies or classifies their evolvability and their ecosystems. This is essentially a framework by which we could develop a classification system, but we don't have the data yet to actually develop the classification system. So one of the things that we're doing in our Cancer Evolution Center is working with Andrea Soteriba and Trevor Graham to develop a classification system for colorectal cancer. So one tumor might be low in all 4 categories and be sort of like a desert. There's not much going on. There's no immune cells. The tumor cells are not turning over very much. There's not much resources, but there's also not much hazards. The opposite end of the spectrum might be more like a rainforest, where you have lots of change over time, lots of diversity of the cancer cells, lots of predators, hazards, but also lots of resources. And you should expect a very different dynamics in those kinds of cancers. So part of the image of the future here is that we should be thinking about how our interventions change the class of the tumor. But it's hard to do that until you actually have a way of classifying the tumor to describe how they're changing. So maybe immune therapy, reduces the diversity in the tumor, but maybe it, and of course increases the hazards to the tumor. But targeted therapy might have different kinds of effects on the classification. And we can start asking, what kinds of tumors respond best to what kinds of interventions. But here's what I think the open questions that derive from this. Well, of course, what are the best predictors for each of these measurements in the ECO and EVO index? What are the best thresholds for classifications? How should we sample the tumors? So one problem we have in general in cancer research, we typically take only one biopsy, and given the spatial heterogeneity of the population, that often is probably not representative of the entire cancer. But then, as I was saying, we should be asking, how do our interventions change the tumor class, and what's the best way of managing each class of cancer? And particularly, I think the question that comes up for us in general is, what if we have the bad case, which I think is often the common case? What if we have a very diverse tumor that's highly evolvable? It's going to respond to our therapy. It's going to evolve resistance, acquired therapeutic resistance. How do we deal with that? And so for the last part of the talk, I wanted to address what I think is a really promising approach, and it actually comes from farming, from agriculture. So the farmers actually are, I would estimate, 2 to 4 decades ahead of us in oncology. The farmers have the same problem. They spray a field with pesticides, they select resistant pests. Now, farmers, from hard experience and theory, assume that resistance is present. Right? I don't think we actually effectively make that assumption in oncology. We hope that it's not present. Farmers assume it's present. They know it's going to be there. About 40, 50 years ago, they tried single drug pesticides. You kill the pest initially, but they come back and you can't control them, they destroy the crops. About 20 to 30 years ago, they did what we do, which is they combine pesticides and multi-drug therapy, right? Just what Larry was talking about. Should work, right? If the chance of resistance to one drug is low, the chance of resistance to 2 drugs should be microscopic, right? Maybe impossible. It doesn't work. It selects for multi-drug therapy. So they learned that failed. That's not an option. And of course, with the rise of environmentalism, they also had a strong pressure to figure out ways of reducing the amount of pesticides. So what they learned was that sensitive pests actually can help them control the resistant pests. And the observation is that to be resistant to a pesticide or to be resistant to a chemotherapy, that comes at some fitness cost. So Bob Gatenby has measured efflux pumps that cause resistance to doxorubicin in MCF7 cells and found that those pumps take up 50% of the ATP of the cell. So half of the entire energy budget of the cell goes to pumping out the drug. So in general, it looks across systems. There's some cost to resistance. So in the presence of the drug, it doesn't matter because they can outcompete the sensitive pests or the sensitive cells because those are dying from the drug. But in the absence of a drug, sensitive cells or sensitive pests have a disadvantage against resistant cells or resistant pests have a disadvantage against the sensitive pests. So what farmers do is they don't spray part of their field. The goal is to keep some sensitive pests alive that can then outcompete the remaining resistant pests. So they will sacrifice a part of their field, and of course, there's struggles over what proportion of the field, 10%, 1%, right? They're willing to take some damage to their crops in order to keep control of the entire system. So Bob Gatenby was inspired by this. He's at the Moffitt Cancer Center to try to change how we manage a tumor, and with the underlying insight that if we can preserve sensitive cells, they can be our ally. So this is the way I think about it. Initially, before we come to treat cells, we have this situation. Our therapy can control or suppress sensitive cells. Sensitive cells can control resistant cells. Standard of care, of course, is high dose, high density therapy. So we kill all the resistant cells. We have nothing left to control the resistant cells. We kill all the sensitive cells. We have nothing left to control the resistant cells. They blow up and kill the patient. And we have no way of controlling that. Right? So we essentially shoot ourselves in the foot by going in with the highest, most aggressive therapy. What adaptive therapy does, this is the name for the protocols that Bob Gatenby invented, essentially is it sets up a negative feedback loop. So if you kill too much of the tumor, if you're killing too many sensitive cells, you need to back off. If the tumor is growing, you raise the dose. If the tumor is shrinking, you back off. Right? That's really counterintuitive for us in cancer therapy, right? If the tumor is responding, we don't tend to want to back off. We think we're doing great. But we're not doing great. We're killing these cells, and these are left behind. Right? It's short-sighted, and that's what Larry was talking about. We can get a short-term response. We can shrink a tumor really well, especially with targeted therapy in personalized medicine, but we suck at the long-term responses, and it's because we're selecting for resistance. So here's the preclinical results. So in 2009, we published this as ovarian model. So immune-compromised mice, the OBCAR3 human ovarian cancer. I believe this is carboplatin. High-dose therapy is this curve right here. So shrink the tumor, but the tumor comes back and kills the mice in a matter of months. Adaptive therapy, where you just adjust the dose to keep the tumor a stable size and keep sensitive cells around, that kept the tumor under control indefinitely. They had to sacrifice the mice for old age. They repeated the experiment, got the same result. Another intriguing result is that the amount of drug necessary to control these tumors decreased over time. That was not predicted by their theory. Not clear why. So we wrote a grant with him to do the follow-up 2 experiments. Does this generalize across to other cancers? So in this case, breast cancer. And can we explain this reduction in the amount of drug you need? So that's 2 different breast cancer models. Again, adaptive therapy. It's called AT1 here. Was able to control the tumors. This was standard chemotherapies, paclitaxel in this case. And it looks like if you look at the tumors at the end of adaptive therapy, they're better perfused than the tumors under standard therapy. So they don't have the areas of necrosis. So it looks like we're getting better drug delivery, and that may be part of the explanation for why you need lower dose. And we also know that low-dose chemotherapy has an anti-angiogenic property, and it's probably pruning down the overly branched and tangled blood vessels that are in a tumor into a more efficient delivery system. So we think that's part of the answer. All right. Then they did the first clinical trial. This is in castration-resistant metastatic prostate cancer. They can measure tumor burden just from PSA levels, so that's how they're monitoring the tumor. They use a slightly different algorithm called AT2. So instead of adjusting the dose over time, you measure PSA levels, and once it falls below 50% of the initial level, you stop dosing. So it's a drug holiday. So it's a fixed dose when you dose, but you go on drug holidays. So it's a pilot trial. So there's only 11 men in the trial. The red bar is when they were on abiraterone, which is the second-line therapy in prostate cancer. Black bars is when they are on drug holiday. Okay. Once the tumor PSA levels comes back up to 100% of the initial level, they start dosing again. So it's on again, off again. Now, one thing I like about this is that it's responsive to the specific dynamics of the men's tumor. So it's a personalized medicine. You can see that some men, like the one on top, have a very slow dynamic. So it took a long time for the tumor to fall below 50% PSA level, and then a long time for it to come back to 100%. Other men's tumors have much faster dynamics, and they're going on and off the drug. But the algorithm accounts for this and adjusts the treatment for the dynamics. It's not a fixed algorithm, right? It's conditional on what the tumor is doing. So in large-scale clinical trials of abiraterone, the standard treatment is just constant dose, which is like these cases here. The median progression time is 16.5 months. They published this when only one of the men had progressed on scan. And if all the other ones progressed the day it was published, they would have been at a median of 27 months. Okay. This is a comparison group of men also at Moffitt Cancer Center on standard of therapy. And you can see that most of them had progressed, and the p-value just between the small groups is so dramatic because the effect is so dramatic that the p-value is tiny. Another nice thing to note is that these men had gotten a average of 47% less drug, 47% of the total drug that these men had been on, because you're going on these drug holidays. So we can reduce toxicity as well. So the reason I'm excited about this, really excited about this, is that for the first time we actually have an algorithm, a method for dealing with the therapeutic resistance, with the evolution of therapeutic resistance. It uses ideas that have come from agriculture where they have the same problem of evolution of resistance, which has been, so it's been proved in that realm. And now we have a way, there's nothing specific about prostate cancer here, right? This should be effective in essentially any cancer and essentially with any drug, as long as you can get cell killing. Now there's important practical aspects to it, which is you have to monitor what's happening in the tumor. In integrated pest management, they check their fields every day for what's happening and adjust what they're doing. Right? In oncology, we dose people with a fixed protocol, often for a number of weeks, sometimes months, and then we don't check what happened until months later. Right? We essentially fly blind. We don't respond to what the tumor does until months later. It's an open question how often we'd have to check the tumor to adjust our doses. We don't know essentially the kinetics. Clearly, prostate cancer has slower kinetics than many other cancers. But those are important open questions. So at Moffitt Cancer Center, they're opening trials in ovarian cancer, melanoma, and thyroid cancer, which are all cases where there's a blood test. At the moment, it's too expensive to do the full body MRIs that one might want to do. We're hoping that we can use cell-free DNA in the blood as a general way of doing this across cancers, even when there isn't a standard antibody blood test. So my vision of the future is that part of our path reports would be including an EVO index or an ECO index to classify the kind of evolution this tumor is going through, not just what mutations it has, not just what phenotype it has. And then we can develop our clinical management plans based on those classes. And of course, incorporate them into clinical trials. You could ask, well, does a highly evolvable tumor respond best to immunotherapy or to targeted therapy, or whatever it is? But the approach of adaptive therapy, where you're keeping sensitive cells alive, is a fundamental change in how we deal with cancer. You would be living with cancer, but not dying from it, right? It's saying we shouldn't be going for the cure, because when you go for the cure, you shoot yourself in the foot, you destroy the very thing that allows you to control the tumor, and then patients in general die. Now, in a very homogeneous tumor with very low evolvability, it's probably the right thing to do to go with a highly aggressive high dose, because the chance that there's resistant cells in those tumors is much lower than the chance there's resistant cells in a highly diverse tumor. So once we have these measurements, these classifications, I think it can guide us in the kinds of therapy we should be doing. But I think in many of our cancers, and most of our cancers even, we're going to have to manage them. And it's just going to be a normal part of old age that we will have cancers that we'll live with, and we'll treat it like diabetes. It's something you monitor, you keep control of. We care for our patients. And in fact, the hints are that we can use lower doses and actually reduce the toxicity and increase quality of life, not only survival time. But we need a common language and a set of tools in order to give us these categories and to monitor the tumors. So this is the reference in case you're interested about the consensus statement from this group about classifying cancers. It was published in Nature Reviews Cancer. And I wanted to acknowledge the wonderful colleagues I've had to develop this work and the funding from the NCI and DoD. Thank you. Yeah.
I was just wondering how the immune system interaction may be changing by that going back and forth, rather than just sensitive cells there, what the impact it's going to have on the environment.
Yeah, it's an important and totally open question. So all the preclinical models were with immune-compromised mice and human cancers put into them. So we have no preclinical data on how the immune system is going to change under this kind of treatment. I think it's also important because we'll be, in general, reducing the doses, so we won't be ablating the immune system so much. So I think the immune system is going to be a big player in these dynamics. But yeah, it's a totally open question, so we need to look at that.
So in the adaptive model, so that's sort of a temporal change.
Yeah.
And going back to Larry's talk, so the patients are dying of the mets, really. Mm-hmm. So would you want to be then trying to kill the mets, hoping to resensitize? So don't treat the primary, treat the mets. And hope that most of the eyes might impact the primary reception.
Yeah, so I think that's attractive. I mean, as an evolutionary biologist, part of the way I think about adaptive therapy is you punish the tumor when it's growing and you let it alone when it's under control, right? So you're putting a selective pressure against the thing that's hard or deadly for us in some sense, and selecting for something that we can live with. And so your idea of kill the mets, but leave the primary tumor alone would sort of select for cancers to stay as localized tumors, which again is something, as we know as clinicians, we can control better. How to do that? Hard, unclear, but yeah, that would be a good strategy to try to look at. There's another question. Yeah.
What happens to the men in the prostate trial who want the adaptive arm after?
Yeah, so I haven't checked with Bob in the last few months, but as of about 3 months ago, it was still only that one had progressed. So they're out somewhere around 38 months of, if all of them progressed today, that day, they would have been at 30 months mean, or 38-month mean survival. So yeah, incredible, really. And so that Dr. Zhang, who was the lead author in that paper, has changed his clinical practice based on that small pilot trial. But of course, this needs to be tested in a large trial. But it's yeah, I'm really excited about it.
Are they continuing under the same protocol?
I believe so. Yeah, they've expanded that one. But of course, clinical management of prostate cancer is changing as well. So they're looking at abiraterone being used as a frontline therapy as well. I think it's becoming the standard of care.
Okay, thank you.